We're Group 17 and our topic is on fluorescent probes. We'll first talk about the types of fluorescent probes in the market. There are various types available that meet a wide range of needs such as organic dyes, biological fluorophores, and quantum dots. Organic dyes are used primarily for bioconjugation of antibodies or avidin because these are small molecules that don't interact with biological function. For biological fluorophores, the most common protein fluorophore used is JFP. Benefits include introduction to bacteria, cells, tissues, and the ability to express the fluorophore either alone or fused with the protein of interest. A few cons include potential changes in the protein of interest, high expression of fluorescent protein that can generate radical oxygen species and in turn create toxicity, and also less sensitivity and photostability than organic dyes. Quantum dots are nanocrystal semiconductors that produce light with tight control of the spectral characteristics. The type of nanocrystals change the emitted fluorescence wavelength, therefore one type of quantum dot could produce a large variety of visible light. Fluorescent works by emitting a source of excitation to the fluorophore, where an electron will move to an excited state, lose energy based on non-radiative decay, then return to its original ground state while releasing a photon. Non-radiative decay is what causes these probes to emit a light at different wavelengths than the absorbed light. Most fluorescent probes will emit light in or near the visible light spectrum. Fluorescent intensity or brightness stays constant between the absorbed and emitted light. Here's a list of organic and protein fluorescent probes grouped by the light that they emit. Choosing the right fluorophore is dependent on the type of biological study performed. However, qualities of a fluorophore to keep in mind are the brightness, lifetime photostability, and absorbed and emitted wavelength. There are two forms of fluorescent detection, microscopy and flow cytometry. For microscopy, something like an antibody or aptamer is labeled with fluorescent probe, which is then excited by UV blue light from the microscope's excitation filter to excite electrons. Once electron relaxes to a lower energy state, green light emits and the microscope detects and visualizes the resulting image. For flow cytometry, a sample of interest is labeled with fluorescent antibodies of dyes and cells from the sample are suspended singly into a stream of fluid. In this single file line of cells, they are excited by a single or series of lasers and is detected and analyzed by a computer. Detectors measure through various physical and chemical. Fluorescent resonance energy transfer, otherwise known as FRET, is used to study the interactions between proteins, nucleic acid, and other biomolecules. The mechanism itself works by energy transfer from one closely spaced fluorophore to another via dipole-dipole interactions. A donor fluorophore overlaps with an acceptor fluorophore and thus starts an energy transition in the acceptor fluorophore. By labeling which proteins or fluorophores are donor or acceptor, scientists may learn more about the molecular interactions, conformational changes, or enzymatic activities. There are various ways that fluorescent probes can be incorporated to study some change at a chemical level. An example of fluorescent tags can be added to a protein of interest. Here, the molecule containing the fluorophore also contains a quencher that prohibits the fluorophore from emitting light. Methods such as FRET, contact, and collision quenching could cause the expected fluorophore to not emit any light. Other ways fluorescent probes can be made to produce light could be due to environmental factors such as changes in pH or temperature. Immunofluorescence is an important immunochemical technique that allows detection and localization of a wide variety of antigens in a multitude of tissues of various cell preparations. Various types of immunofluorescence procedures are available, but the gist of it is that it produces light when an antibody binds to the target antigen. Monoclonal antibodies, only one type of antibody is used conjugated with a fluorophore that activates when the antibody binds to the target antigen. For indirect polyclonal antibodies, multiple types of antibodies are used to bind to the primary antibody. The antibody binds to the target antigen. Each of the secondary antibodies are conjugated with fluorophores to amplify the fluorescent signal. For indirect monoclonal multi-conjugated antibodies, the second antibody binds and contains multiple complexes that are conjugated with a fluorophore, and in turn produces greater amplification. For indirect polyclonal multi-conjugated antibodies, similarly to the indirect monoclonal multi-conjugated antibodies, except multiple secondary antibodies are introduced. This offers the greatest amount of signal amplification.
Fluorescent probes are making their way into clinical use for diagnosis and imaging. The following are two that are FDA approved. Methylene blue has been around for many years, but its fluorescent properties haven't yet been used at their full advantage. Although commonly used as a stain in microbiology studies, it also exhibits an emission spectrum at near-infrared region. This makes methylene blue a probe of interest for deep tissue imaging. Near-infrared light is more likely to pass through layers of skin, which is why it is used for the visualization of ureters, glands, tumors, and more. In the case of imaging ureters, methylene blue prevented surgeon-caused injury by illuminating this duct and preventing serious complications. Other case studies of methylene blue in patients show the detection of the parathyroid so that it may be avoided during surgery. In the past, methylene blue as a stain was used to color the parathyroid but required high doses of the organic dye. This led to high frequencies of secondary effects. However, the near infrared emission of methylene blue can be observed even at lower dosages and therefore preventing the high frequency of mild secondary effects and almost eliminating any serious effects. Because of these benefits, methylene blue is one of the 9 approved parathyroid identification techniques in the market. As previously mentioned, there are secondary effects on patients who received an intravenous dose of methylene blue. However, the seriousness and frequency of these effects are diminished when doses are reduced to under 2 milligrams per kilogram. In the list of possible symptoms, oxygen desaturation is listed. However, this is only because methylene blue interferes with the measurement tool, producing a false positive for oxygen desaturation. At higher doses, serious complications can occur. In particular, patients taking SSRIs and SNRIs are at risk of developing serotonin syndrome after use of methylene blue during surgery. It should be also avoided in patients with G6PD deficiency and pregnant women. The first FDA approved fluorophore is indocyanine green, primarily used for surgery and detection of cancer. Indocyanine green accumulates within the lymphatic system and it is used to detect the nearest lymph nodes to a tumor. The accumulation of indocyanine green occurs due to a binding to beta lipoproteins. The detection and then removal of these lymph nodes is vital in preventing a reoccurrence of cancer in breast cancer patients. Additionally, indocyanine green is a cheaper and safer alternative to the radioactive technetium isotope previously used. In two meta-analyses of 2,500 and 1,700 surgeries, indocyanine green was more effective at physicians detecting sentinel lymphatic nodes. Although some side effects may be present after intravenous injection of indocyanine green, these occur at very low frequencies between patients, and it's considered more safe than the previously mentioned methylene blue. Shown on the screen is a list of either groupings or specific fluorophores that are either already FDA approved or in preclinical studies with a summary of their applications.
Fluorescent molecules have many uses outside of just the medical field and medicine in general. Quantum dots are utilized in solar panels, fluorescent LED lamps are commonly used around homes, and the way that fluorescent and LED lamps work is that the fluorescent coatings convert the UV or blue light into longer wavelengths, which in turn creates white light. Even childhood toys and glow in the dark comes from the concept of fluorescence. Another popular use in the clothes and textile industry is fluorescent colors. The glowing effect is produced by absorbing the light energy or radiations that fall into a narrow band of wavelength and emitting the light energy with comparatively longer wavelengths.